Okay, I'm curious, how many of you have started working on your cheat sheet, if you want to call it that, for your exam? You really should be, because what I think would be a really productive way to approach this is start making at least the skeleton of that cheat sheet. Any idea why I decided on front and back of 2 pieces of paper? From an organization, well, there's 4 chapters, right? And so that might be one way to start organizing your information. Now remember, chapter 17 is bonus, so you may choose or not to choose to put stuff on there about virus, but gives you a way to start breaking information down. But what I would do is I would start going through your PowerPoints in the textbook and saying, I know I want these diagrams on here, okay? Put those on there. Start going through the bold-faced terms and the vocabulary terms and say, these are things I have trouble remembering. I'm going to put those on there. Then once you've got that, as you start doing study guide questions or start reviewing your notes or going through old exams or whatever strategy you use to start preparing for exams, have those things sitting there, okay? And then you might find out, for example, that on some of the diagrams, for example, there's some additional information you want to add. We were going over with somebody yesterday the lac operon, and the diagram in the textbook doesn't make reference to structural genes, and yet some of the study guide questions ask you about structural genes. So you might want to add that on that particular little diagram. But these notes can kind of evolve as you study. If you use them while you're studying, then you're going to know where stuff is in your notes. And then that way, it's not going to take you long if you say, oh, this question deals with this particular thing, and I know where it is in my notes. If you don't know that, you can quickly go to those notes. But you're not going to have time on the exam to just constantly be referring back and back to your notes, or you're not going to finish in time, okay? So I think the main utility in these is going to be preparation of those notes. That's what's really going to help you study. And then they'll provide a little bit of a crutch, so there's all this stuff you don't have to just flat out memorize. I'm hoping that this will give you an opportunity to be able to look at big picture a little bit and understand how these things work. But it's not going to do you any good to have a picture of the lac operon in your notes if you can't then go through that and say, this is how this works. Okay? So this is just kind of removing the burden of having to memorize all this stuff and kind of forgetting some of the key elements. It'll be there for your reference. So I would make it a priority to start working on these and start getting them organized as part of your study. Okay? As far as today's information is concerned, this falls into the category of bonus questions on your next exam. If you choose not to put a lot of effort into reviewing this information for the exam, please pay attention for the next 2 days while we talk about it in class, because hopefully some of it will stick, and that may result in you being able to answer some of these bonus questions, even if you don't really put any kind of priority in terms of kind of studying that information again for the 4th exam. Okay? Any questions? Okay, so I have some questions for you then. We have talked about virus a little bit already in a couple of contexts, and you've all heard about virus before. So let's start out by just making a list of some of the things about what do you know already about virus.
They reproduce on their own. They have to have another organism.
Okay, so they can only reproduce inside of another cell. One of the words that we use oftentimes to characterize a virus is that it is an obligate intracellular parasite. Okay? We all know what a parasite is, right? Something that lives at the expense of another organism. Intracellular means it has to live inside of a living cell. Does anybody know what obligate means? Obligate means strict or absolute. And so these are the only conditions under which these viruses can, quote unquote, live. Okay? What kind of things do they have to parasitize from the cell? What does a virus have to get from the cell? What are viruses made of?
Proteins and nucleic acids.
Okay, viruses are only made of two components. Proteins and nucleic acids. Okay? Are they alive? No, they have some of the properties of life. What's the major property of life that viruses have? What property of life do they share with all other living organisms?
They have genetic material.
They have genetic material, yes. What else?
You need to multiply.
They can multiply or reproduce, right? But they're limited. They can only multiply or reproduce in a living cell. When they're outside of a living cell, viruses are just inert chemicals, proteins and nucleic acids. And as a matter of fact, it really surprised people in the 1950s when they were able to actually crystallize viruses. Normally, homogeneous mixtures of molecules, like cells, can't be crystallized. But viruses can be turned into crystals. Okay? Do viruses metabolize? So they have metabolic biochemical pathways, like glycolysis. Citric acid cycle. You're right, Antonio. No, they don't. Okay? So one of the things that they have to obtain from cells, one of the things they parasitize, is energy. Can viruses make their own molecules? This is a little bit of a loaded question. The answer is kind of yes and no. Can they make their own proteins? Not really. They actually use the cell's genetic machinery to make proteins. Viruses don't have ribosomes, for example. Didn't we say at the beginning of the semester that one of the requirements for being a cell was to have ribosomes so you could make proteins? Bacterial cells have them. All cells have ribosomes. Viruses don't. What else do you know about viruses? That's great.
You can make vaccines for viruses.
Okay. You can make vaccines against virus. Why is that important? Why do you vaccinate?
So your body will make antibodies to fight against it.
Okay. You're trying to prevent getting sick, right? You're trying to prevent getting viral diseases. So, can virus cause diseases? Yeah. Anybody know an example of some viral diseases? Influenza. And influenza is interesting. A lot of people, lay people, have this misconception about the flu. And you hear people sometimes referred to as, having the stomach flu. Okay? Stomach flu really isn't influenza. Influenza is a respiratory disease. And it can be a lethal disease. There are all kinds of different types of influenza. We'll talk about influenza virus a little bit. What other diseases can virus cause besides influenza? Okay. Chickenpox?
HIV.
HIV? Pardon me? Ebola? Common cold? Smallpox? One viral disease that we really have eradicated, although we do have some samples of smallpox virus kept for research.
Polio.
Polio? Most of you don't hear about polio anymore. How come? Polio has been fairly well eradicated in part due to vaccination. When Dr. Morton and I were growing up, polio vaccine was just first being developed. And polio was a real scourge in the time prior to that. Have you ever seen pictures of people living in iron lungs, for example, where polio virus has paralyzed their muscles, including muscles that allow them to breathe? And so you had to live in this big iron lung which ventilated you and allowed you to breathe. Pretty grim existence, right? Sometimes people that recovered suffered permanent paralysis from polio. There's one of our more famous politicians who suffered from polio. Really see if you can know your history here. FDR, yes. Franklin Delano Roosevelt, 4-time president. Okay. So there's a lot of viral diseases out there, and fortunately, we have been able to develop vaccines against many of these particular diseases, and Brittany's correct. What the vaccine does is it basically fools your immune system into thinking you have been exposed to the real disease. And when that happens, you start producing antibodies. And fortunately, your immune system has a memory. And then if you're exposed to the real virus later on, your immune system remembers that prior exposure, and you can respond very quickly, and oftentimes you don't even suffer any symptoms. So that's another thing about diseases, is that most viral diseases have some associated symptoms, and those symptoms are oftentimes used in diagnosis of what kind of disease you have. What else do you know about virus? What is the genetic material, the master set of instructions for all living cells? DNA, right? And the DNA is in a double helix, and so it is double-stranded DNA. Is double-stranded DNA the genetic information for virus? It can be. There are some virus whose genome is made up of double-stranded DNA. But there are other types of viral genomes as well. Some virus have a single strand of DNA as their genetic information. Some virus have double-stranded RNA as their genetic information. And some virus have single-stranded RNA as their genetic information. So there's a lot more variability in virus in terms of what is the stuff that provides the genetic information for this thing we call the virus. So this actually is kind of a convenient way to start breaking virus down into kind of families or groups. The double-stranded DNA viruses, the single-stranded RNA viruses, etc. What if you get sick with a viral disease? What treatment options are available for you? Okay, there are some antiviral drugs. Do those include antibiotics? Nope. How come? Why would antibiotics not that virus? Okay. Yeah, those are for bacterial infections. Antibiotics typically fall into a couple of main categories. There are those antibiotics that interfere with cell wall synthesis, bacterial cell wall synthesis. Viruses don't have a cell wall. We don't have a cell wall. So that's one of the reasons why antibiotics can be used to selectively kill bacteria but don't kill our cells. What's the other major group of antibiotics? What's their target? They target ribosomes, and specifically 70S ribosomes, those bacterial ribosomes. What are our ribosomes? 80. Okay. So again, that gives us a basis for selectivity. But virus use human ribosomes to make their proteins. So the antibiotic isn't going to help. The antiviral drugs, we're finding more and more of these all the time. Some of the antiviral drugs interfere with DNA replication. One of the first antivirals was a drug used to treat AIDS called AZT, azithromycin. But we're finding some other interesting antiviral drugs as well that target other aspects of viral. There are some antiviral drugs that are neuraminidase inhibitors. Don't worry about that word. Neuraminidase is a viral structure. It's a protein that's involved in attachment of virus to cells. And if you can block these neuraminidases, it can help to prevent virus infecting cells. There are some enzymes called proteases that are necessary for the completion of the virus life cycle. And protease inhibitors can interfere with viral replication. One of the things that we're starting to find is that many times mixing several of these drugs into what we call a cocktail is much more effective than using individual drugs to try to treat viral infections. So the HIV treatment now is typically a triple cocktail of 3 different drugs. And unfortunately, that makes it pretty expensive. And there are all these really kind of demanding dosing requirements where you've got to take this pill at this time, this pill at this time, and if you miss a dose, it really screws things up. So that's one of the reasons why in populations where there are people who have less education, it's more difficult to have effective HIV therapy, and also the cost is a real issue. But we are making some advancements on antiviral drugs. For influenza, you may have heard of Tamiflu before. One of the things about these drugs many times, however, is that you've got to diagnose the infection fairly early on. If you don't start taking Tamiflu within about 2 or 3 days of your initial infection, it just really doesn't do much good. So this idea of diagnosing the disease properly is really important in treatment of viral diseases. So what does a virus do when it gets its genetic information into a cell? Because remember, that's what Hershey and Chase demonstrated, didn't they? With that P32S35 experiment, was they showed that genetic information entered the cell. And in the case of the virus they studied, the T phage, they were able to demonstrate that it was the nucleic acid that got into the cell. But what's the consequence of viral genes getting into your cell? Why is that bad? What is the consequence of you opening an attachment on your email that contains a virus?
You get infected.
Yeah, your computer gets infected, and the malicious code can basically, rewrite programs in your computer. And one of the programs that it sometimes can rewrite is it can kind of co-opt your contacts. And so it can cause your computer to send out emails also containing that malicious code. And so that particular code propagates and can be sent to other computers. A lot of analogies between computer viruses and cellular viruses, and both can be very damaging and deadly. What the viral genes really do is they really reprogram the cell. And the cell starts preferentially using viral instructions to do what? to make viral proteins and viral nucleic acids. And so basically, the intent of the cell is completely subverted. And the cell is changed into, let's become a virus factory. And let's start making just virus parts. And then they're able to come and assemble together. The cell fills up with virus. And then eventually the virus are released from the cell. And each one of those released virus can infect another cell. And then those cells become infected and produce multiple virus. And so you get kind of a chain reaction where more and more cells are infected the longer the viral infection rages. And there can be real consequences from that. There can be disease symptoms. There can be death in the case of virus like Ebola. Pretty nasty disease. One of the characteristics we haven't talked about is that viruses are typically small. And really, practically, we can only see virus with an electron microscope. Electron microscopes weren't developed until 1950s. So prior to that, people really were certain a virus existed, but we couldn't see them. They can be really, really tiny. There are viruses that are smaller than a ribosome. Some viruses can be relatively large and approaching the size where you could potentially even see them with the standard light microscope. But that's really the rule, the exception rather than the rule. Most viruses are quite small and quite tiny. And this kind of gives you a representation of relative sizes of a bacterium, a human cell, and in this case a T-phage virus. So here's the nucleus of our human cell. Here's the cytoplasm. Here's a bacterium, okay, and then you can see the relative size of the virus. What kind of cells are prone to viral infection? We definitely know human cells, right? How about a reptile? Do reptiles get viral infections? How about birds? How about fish? How about bacteria? Do you remember what we call a virus that infects a bacteria? It's called a phage, or phage, some people say. So anytime you see this word, phage, you should think of this as a bacterial virus. Plants, can they be infected by virus? Yeah. Fungi? Yeah. Algae? Paramecium? Yep. All living cells can be infected by virus. Can all virus infect all cells? No. That's another characteristic of viruses. There's some specificity. Sometimes the specificity is at the species level. So a bird virus might not be able to infect a human virus. Likewise, a human virus might not be able to affect a bird. Sometimes even within species, there are different levels of specificity. There are certain tissues in the human body, for example, that are prone to viral infection and others that are not to the same virus. If I gave you a vial of cold virus and you rubbed it all over your skin, would you get a cold? No. But if you then touched your nose or your eyes and those mucous membranes became exposed to the virus, would you get a cold? Yeah. That's why hand washing is so important during cold season. Because you want to make sure you don't inadvertently pick up virus from someone else and then touch your nose, your mouth, your eyes, because those tissues are very prone to be infected by those cold viruses. So specificity is very important. Now, unfortunately, what happens sometimes is that virus mutate. And sometimes virus that previously had only been able to infect a pig, they're called porcine viruses, or birds, avian viruses, sometimes mutate and are able to cross species barriers and now infect humans. And that's been particularly the case in influenza. So some of our more nasty influenza outbreaks have involved avian viruses that have now crossed the species barrier and been able to infect humans. Or viruses that only infected swine and now can infect humans. Swine flu, okay? Viruses aren't alive. They're in that kind of netherworld between living and non-living. They are obligate intercellular parasites. And when they infect, they typically will turn a cell into a virus factory. Now, I've got to hedge a little bit on that particular statement because we're going to see later on that there are a couple of different kinds of life cycle of virus. And this statement only applies to one of the two types of life cycles. But we'll get there in a minute. Okay. So they only consist of protein and nucleic acid, either DNA or RNA, but not both. Okay, our cells have DNA and RNA. Virus, if they have DNA, no RNA, and vice versa. Talked about the nucleic acid makeup. The nucleic acid makes up a region of the virus called the core. And the core is then surrounded by a protein shell, if you like. So if you want to think of a virus as an M&M, or a Skittle, okay? The chocolate in the middle of the M&M is the core. And in the colored candy shell is the coat, or capsid. So this capsid is a bunch of proteins organized together to form this viral shell. How those proteins are organized determines the shape of the capsid. And the shape of the capsid defines the shape of the virus. We'll take a look at some capsid shapes in just a second. Now, some viruses do have a structure called an envelope. And this is really essentially a membrane that surrounds the capsid. But that membrane comes from the cell that the virus infects. Okay? Now, sometimes there are proteins in the membrane that are viral proteins, but definitely the phospholipid that makes up this envelope is host cell, infected cell in origin. And then some virus, including the HIV virus, have packaged up inside their capsid some enzymes in addition to the nucleic acid. And that's particularly true with one type of enzyme that we've talked about a little bit already, the reverse transcriptase enzyme in retrovirus. You remember what reverse transcriptase enzyme does? Copies RNA into DNA. Okay? That's kind of the opposite direction of the normal central dogma. Okay, so here is an illustration of what a capsid of a particular type of virus might look like. And this is an electron micrograph of that particular virus. So this is a plant virus called tobacco mosaic virus. This is one of the first plant viruses that was really extensively studied. And it causes the leaves of tobacco plants to become all mottled. They have yellow splotches all over them. And this really does have an effect on those tobacco plants' growth. And if you get a heavy viral infection, it can kill the plant. So what's the virus consist of? Well, in the middle of this tube right here, which is the capsid, wound in the middle of that is the core of single-stranded RNA. So that capsid shell kind of protects the genetic information. Each one of these little bumps right here is a polypeptide chain folded up into a three-dimensional shape. And those individual subunits or polypeptide chains are called a capsomer. So basically we build this tube out of all these little blocks of protein. So this is kind of like the tube that's inside toilet paper rolls and paper towel rolls, okay? And then packed inside of that is the genetic information. So that's a pretty simplistic viral morphology. Here's one that's a little bit more interesting looking and a little bit more complicated. Okay? This is a type of virus called an adenovirus, and this causes some respiratory infections. So, here's our core right here of DNA in this particular case. And then here are the capsomeres, so these are proteins that are all stacked together to form this icosahedral arrangement. It's got kind of angles to it, okay? And then actually inserted into this capsid are some modified proteins, glycoproteins. They're sugar-containing proteins. So this again is the electron micrograph. So this looks very different than the tobacco mosaic virus that we just saw. Here's the guy we've seen before. This is a T phage. This is the Hershey and Chase virus that was used to determine the DNA was the genetic information. And again, this has got a fairly complex morphology. So this whole thing right here represents the capsid. And then packed up in this region of the capsid, in the head region, is the genetic information. But then we've got this, tail fibers here and this little collar. Several different proteins make up this particular capsid. And there's again is our electron micrograph of that T phage, that bacterial virus. There's another electron micrograph of the T phage. Here's influenza virus. Okay. Here we can see that the genetic information of the influenza virus consists of multiple copies, not a single copy now, but multiple copies of RNA. So this is an RNA virus, but there are more than one copy of that set of genes packed into this virus. Surrounding that is a capsid, so each one of those is surrounded by a capsid. And then this whole group of capsids are surrounded by a piece of cell membrane. This is the envelope. And then stuck into this membrane are various glycoproteins. And these are actually viral in nature. Those proteins were coded for by these viral genes. This is getting to be a fairly large virus, and this is an electron micrograph of the influenza virus. Any idea what these little glycoprotein spikes do?
Attached to cells.
They attach to cells. Okay, so one of the things that has to happen for virus to infect cells is the first thing that has to happen is attachment. And if the virus can't attach to the cells, it can't infect it. So these little spikes don't match receptors on your skin cells. But they do match receptors on your mucous membranes that are part of your nasal cavity. So they can infect those cells. Here's an enveloped virus. This is the HIV virus. And the little wrinkle that this shows here is a couple of things. So here's our capsid surrounding our two molecules of RNA. And then we've got our membrane, which comes from the host cell. We've got some glycoproteins here. And then these little green balls right here represent viral enzymes that are actually part of the virus structure. Those are reverse transcriptase enzymes. Virus has to have them because the cells they infect don't have those enzymes. This is just to show you that we can start breaking down virus into groups based upon the nature of their genetic information. So we've got double-stranded DNAs, single-stranded, double-stranded RNAs. And then single-stranded RNAs are oftentimes broken down into subcategories. Don't worry about any of that. But again, look at some of the virus here that you might recognize from a disease standpoint. Herpes, for example. Papilloma, HPV. Diarrhea, polio, cold, yellow fever, rabies, measles, mumps. Okay, so there's a long, exhaustive list of diseases that virus can cause in humans, and there's an equally long list of diseases that virus can cause in plants. They're kind of scary things. They are typically host-specific, although we did talk about the fact that viral genes can mutate, and sometimes virus can then cross those species barrier. And that's particularly the case in these influenza virus. That's why they're so scary. There's another nice illustration of a bacterial cell. I believe this is an E. coli. And you can see that multiple phage have attached to this cell and are probably in the process of injecting their DNA into the cytoplasm. That DNA is going to sabotage the cell, kind of a terrorist, reprogram the cell, and turn it into a virus-producing factory. It's hard to generalize, but a good kind of ballpark estimate is that when a cell gets about 200 to 300 virus built within it, then that typically will result in the cell bursting. That's also called lysis. And then each one of those 300 virus that are released could infect a new cell. So you can see how a viral infection can really pick up steam pretty fast. Originally, when the viral infection just started, you probably don't suffer that many symptoms. But then pretty soon, as more and more cells become infected, the symptoms become really overt and very easily identified. We talked about some of the diseases that they cause. Here is a tobacco mosaic virus causing this mottled appearance on the leaves. A lot of chlorophyll is destroyed as a result of these particular infections. This is a SARS virus. I don't know, Larry, you know how long it's been since we had that SARS outbreak? Was that?
Probably 10 years.
Yeah, probably at least 10 years ago. It was had its origins in the Far East, and people were very commonly wearing masks to try to prevent inhaling the SARS virus. This is a micrograph of this SARS virus. It's a type of virus called the coronavirus because of these glycoprotein spikes sticking out from the envelope. Kind of looks like a crown. Here is a influenza virus from the 2009 H1N1 pandemic. We'll talk a little bit more about what those designations mean later on. And here are people at an airport. What are they screening them for? Why do they have to walk past this before they get on a plane? That can't see the virus, can it?
Is it to see like the heat from their face?
Yeah, it's a thermal imager, and they want to see if they have a fever. And then people with fevers weren't allowed to get on airplanes. Okay, we're going to talk about two types of life cycles. And probably the only one we'll get through today is the more general kind of life cycle, which is the virus lytic life cycle. As a result of this life cycle, a cell is infected, virus are produced, and the cell bursts and releases new virus. And that's why it's called a lytic cycle, because the cells lyse. Okay? Typically, any type of cell can be infected by a virus and go through this lytic cycle. And it doesn't take very long. Typically 20 to 30 minutes to complete the whole life cycle. It's pretty fast. Here are the generalized steps of the lytic life cycle. And each step follows the other. And if one of these steps doesn't happen, the rest of them don't happen. So the first critical step is attachment. Some kind of molecule on the surface of the virus. It might be a capsid protein. It might be an envelope protein. Has to bind to a receptor on a cell. That's attachment. So in the case of analogy of your computer viruses, attachment is the kind of same as receiving that infected email. Is your computer going to get infected if you don't open that email? No, shouldn't. Okay. Just like a cell's not going to get infected unless that viral genetic information gets into the cell. In attachment, it's still outside the cell. So it's got to get into the cell. How does that happen? Well, in the case of that T phage, the virus is kind of like a hypodermic syringe. And it actually contracts and punches a hole in the membrane and forces the nucleic acid into the cell. Not all viruses do that. Many viruses are brought inside by endocytosis. So the cell membrane pinches in and brings in that virus, like it would a food particle. Now it's inside of a vacuole. And if that vacuole fuses with a lysosome, remember what lysosomes are? What are the lysosomes at? Digestive enzymes that can digest away that capsid. That process of digesting away the capsid is called uncoating. And what's that analogous to? Opening up that email. Now your computer's infected with bad code. Now your cell is infected with bad code. What happens next? Genetic information reprograms the cell. And it starts causing it to produce new viral genetic information, more RNA or more DNA, depending on the virus, and also viral proteins. So the cell starts filling up with these viral components. At some point in time, they come together, and the viral proteins start surrounding the genetic information. A capsid forms around the core. And now we have a new virus particle. What's interesting about this process of assembly is that it's spontaneous. It happens on its own. Remember I told you that there was another situation in our normal cells where proteins and nucleic acids spontaneously come together to form something in the cell that's necessary for the cell? What am I talking about? Ribosomes, remember? Transcribe genes for ribosomal RNA. Translate proteins for ribosomal RNA. And then those spontaneously come together to form the big and the small subunit. That's called self-assembly. Okay? And then when the cell fills up with these virus, and again, somewhere in the vicinity of 200 to 300, each virus has what is called the characteristic burst size, where there's a certain number of virus that are formed before the cell bursts. And then finally, virus are released. So, here is a illustration of a lytic life cycle of an animal virus, probably in this case. Here's the capsid. Here's our core. And this virus is brought into the cell, probably by endocytosis. The viral genetic information is used to make more proteins and more viral genetic information. Those come together and form virus particles, and then the cell bursts and releases those virus so they can infect new cells. Envelope virus have a very similar type of life cycle, but there are a couple of little wrinkles in their life cycle that we'll pick up with next time. I think this is a good place to stop. So tomorrow we will finish talking about virus, and then remember what's happening next week. Next week we have got Monday, you're going to do some lab stuff, is that correct? There will also be some time, if we have it, to review. Those of you who want to take the quiz, and why in the world wouldn't you take the quiz? Because you can drop it. We'll take the quiz. Tuesday exam. Next Thursday and Friday, review for the final. Did you have anything you needed to say to lab people about lab today?
Oh, yes.
Oh, yes.
Those of you who are in lab this afternoon, you will have